What you're listening to is the audio version of an article in the Life Extension Magazine 2025 edition. The title is Slow Biological Aging and Increase Health Span by Michael Downey. Here's a quick summary in case you don't have time to listen to the whole article. An extract of the Scarlet Beebalm plant addresses 5 of the known mechanisms or hallmarks of aging based on cell-based studies. In a recent human trial, those who took Scarlet Beebalm extract showed no significant epigenetic aging during the study, while a placebo group aged epigenetically by 1.7 years. This extract also significantly lengthened telomeres, which are the protective caps on chromosomes, compared to a placebo, and improved quality of life. By targeting multiple drivers of aging, this plant extract may slow biological aging and lead to longer health span, years of healthy life. Scientists have identified 12 hallmarks of aging that contribute to degenerative processes and the risk of age-related disorders. A plant extract, called Scarlet Beebalm, has been found that targets 5 of the recognized contributors to aging. In preclinical work, this extract was shown to reduce telomere shortening, oxidative damage, genomic instability, cellular senescence, and chronic inflammation. In a clinical study published in 2025, adults taking this extract for 12 weeks had no significant increase in epigenetic age, a major determinant of biological age. The epigenetic age of placebo recipients, on the other hand, increased 1.7 years. You don't want an older epigenetic age. Compared to placebo, the extract led to improvements in leukocyte telomere length and stabilization of DNA methylation age. These results indicate the potential for this extract to slow multiple contributors of biological age and increase health span, the number of years lived in good health. Biological versus chronological aging. Many biological processes are associated with degenerative aging. Scientists have identified 12 of these biological processes that are called hallmarks of aging. Over the years, adverse changes in these processes determine our biological age. This is different from our chronological age, the number of years we have lived. Compared to others of the same chronological age, those with accelerated biological aging may seem more frail, are more likely to develop age-related diseases, and have reduced health span. Researchers have searched for ways to slow the rate of changes within one or more of these hallmarks of aging, hoping to decrease biological aging and increase health span. After testing other candidates, they identified a plant called Scarlet Beebalm with abilities to target 5 of the hallmarks of aging. Scarlet Beebalm is an herb native to North America. It has long been used safely by Native Americans to treat respiratory infections and dental disorders. This herb is abundant with beneficial flavonoids, including didymin. In preclinical studies, didymin is known to boost the function of mitochondria, the energy-generating powerhouses of cells, and of endothelial cells lining the insides of blood vessels. An extract of Scarlet Beebalm, standardized to 4% didymin, has been demonstrated to slow mechanisms of aging. In lab studies, this extract did the following. First, it reduced oxidative stress. The extract reduced markers of oxidative damage in stressed cells and lowered levels of protein carbonylation, a type of damage that occurs in proteins exposed to oxidative stress. Second, it slowed telomere shortening. Telomeres are like protective caps that help shield DNA and keep genetic material stable. Telomere length and, more importantly, the rate of telomere attrition are strong predictors of species lifespan. Telomere shortening is associated with chronic disease, aging, and higher risk of mortality. Cells treated with Scarlet Beebalm extract had a significant reduction in the rate of telomere shortening compared to untreated cells. The third thing it did was protect against DNA damage. In cells exposed to harmful substances or conditions that damage DNA, the extract reduced markers of DNA damage, showing improved DNA repair and genomic stability. The fourth thing it did was decrease cellular senescence. Dysfunctional senescent cells damage tissues and drive aging. Scarlet Beebalm extract decreased a common aging marker in aged human cells, indicating reduced cellular senescence. Finally, the fifth thing it did was it reduced inflammation and improved endothelial function. The extract reduced pro-inflammatory markers in endothelial cells and decreased permeability or leakage in microvascular endothelial cells. Excess permeability can increase risk for tissue damage and disease. Together, these results indicate anti-aging effects and potential cardiovascular benefits. C. elegans are a type of roundworm that live only 2 or 3 weeks, making them ideal to study aging. In a study, worms were divided into 3 groups. One group received no treatment. Another was given sulfamethoxazole, a compound known to extend health and lifespan in C. elegans, and the third group was treated with the Scarlet Beebalm extract. Over 7 days, the Beebalm extract treated worms showed increased time spent moving, improved speed, and greater distances moved. In other words, the extract enhanced physical activity and vitality over a significant period of the worm's lifetimes. In a human study published in 2025, 81 participants aged 45 to 65 took either 100 milligrams of Scarlet Beebalm extract or a placebo daily for 12 weeks. 2 markers of biological aging showed favorable outcomes in those who took the Beebalm extract: DNA methylation and telomere length. DNA methylation is a critical epigenetic process and considered as a predictor of health span. Scientists measure DNA methylation age by analyzing chemical tags called methyl groups attached to DNA. It is often used to help determine biological age. In this study, the placebo group's DNA methylation age increased by 1.7 years in 12 weeks. The Scarlet Beebalm group, on the other hand, showed no significant increase. This stabilization of DNA methylation aging shows support for cellular function and longer health span. Telomere shortening is a biomarker of cellular aging and is associated with age-related disease and mortality. The Beebalm supplemented group showed an increase in leukocyte, a type of immune cell, telomere length, compared to the placebo group, which experienced a decline during the study period. This suggests that Beebalm beneficially lengthened telomeres rather than merely preventing their loss. Slowing DNA methylation aging and reducing telomere shortening may contribute to a longer health span. In addition, according to a quality of life questionnaire, those who took the Beebalm extract engaged in more frequent and vigorous physical activity, suggesting they felt healthier as they aged. Here are the 12 hallmarks of aging, and the first 5 of these are targeted by Scarlet Beebalm, with clinical findings validating 2 of these effects. Okay, for the first one is oxidative damage, the second one is telomere shortening, the third one is genomic instability, the fourth one is cellular senescence, and the fifth one is chronic inflammation, which Beebalm seems to target. The sixth one is loss of proteostasis or protein balance. 7 is disabled autophagy or cellular housekeeping. 8 is deregulated nutrient sensing. 9 is mitochondrial dysfunction. 11 is stem cell exhaustion. 11 is altered intercellular communication. And 12 is dysbiosis or imbalance of gut microbes. So finally, if you want to know the effect of telomere shortening and human mortality, a 2003 study published in The Lancet examined telomere length in a group of individuals aged 60 or older. Those with shorter telomeres had an associated 3.18 times higher mortality rate from heart disease and an 8.54 times higher mortality rate from infectious disease. And finally, here are the references at the end of the article. If you can't read it, maybe you could pause it. If you still can't read it, you can just go to the lifeextension.com website, click on magazine, and go to the December 2025 edition. and go to page 29.